Hey everyone, and welcome back to The Explainer. Today, we're diving straight into a comprehensive guide on Chapter 5 for DSE Biology, Cell Cycle and Division. We're going to take a fascinating, microscopic journey into the exact mechanisms of how life grows and reproduces. We'll be breaking down the fundamental processes of cell division and turning all those dense biological concepts into really clear visual takeaways. So if you've ever wondered how a single, microscopic cell manages to multiply and form incredibly complex organisms, well, you're in exactly the right place. Let's get into it.
Here is our game plan for today. We're going to cover the structure of genetic material, the cell cycle and mitosis, cytokinesis in plants versus animals, the onion root squash experiment, the stages of meiosis, and finally, the grasshopper testis squash. All right, Section 1, Structure of Genetic Material, the building blocks. Before we can really grasp how a cell divides, we need to take a step back and look at the absolute fundamental units of heredity, which are, of course, the chromosomes. Now, a chromosome isn't just some loose strand floating around randomly in the cell. It's a highly organized, thread-like structure. It consists of a long DNA molecule, which carries your actual genetic code, tightly coiling around these specific proteins called histones. And this packaging is absolutely crucial. Think about it. Without it, there'd be no way to fit massive amounts of genetic information into a tiny cellular nucleus. You can really clearly trace that famous DNA double helix right here. Notice how it acts almost like a thread wrapping around those grouped histone proteins, which then supercoil to form that much larger, familiar chromosome structure you see inside the nucleus. It's like wrapping miles of kite string around a spool so it doesn't get tangled. This intricate, highly organized folding is what keeps our entire genetic blueprint intact. Now, when a cell is just resting, this genetic material exists as a fine, basically invisible network called chromatin. But right before the cell divides, things change. The chromosomes replicate. They shorten, they thicken, and suddenly they become totally visible under a microscope as these distinct rod-like structures. What you're seeing here is a replicated chromosome made up of two perfectly identical threads. We call these sister chromatids, and they're pinched together at the central point called the centromere. Moving on to Section 2, Cell Cycle and Mitosis, the chronological flow. Let's map out the orderly sequence of standard cell growth by looking at the timeline of the cell cycle. A really massive takeaway for you here is that cell division itself is actually just a tiny, tiny fraction of a cell's life. The vast majority of its time, we're talking about 90%, is spent in what's called interphase. An interphase is broken down into G1 for active growth, S phase, which is super important because that's where DNA replication occurs to double the genetic material, and then G2 for further energy storage and synthesizing organelles. Only after all that intense prep work is finished does the cell finally enter the M phase, or mitotic division. So let's zoom in on the first half of that mitosis process. On the left side, we start with prophase. The nuclear membrane is actively breaking down, and those sister chromatids we just talked about are condensing into fully visible structures. Then, as things transition to metaphase on the right, something really beautifully organized happens. The paired chromatids line up perfectly, one by one, right in the middle, the equator, of the cell, and they attach to spindle fibers by their centromeres. It honestly looks like microscopic choreography. Continuing with that sequence, anaphase is where the real action happens. The centromeres split. Those spindle fibers physically contract and literally yank the sister chromatids apart, pulling them toward opposite poles of the cell. At this point, they're officially considered independent daughter chromosomes. Finally, we hit telophase. A new nuclear membrane forms around each set of chromosomes at the poles, giving us two identical daughter nuclei. And the chromosomes? They just begin to uncoil right back into that messy chromatin form. Section 3, Cytokinesis, Plants versus Animals. Okay, so now we have two brand new nuclei, but they're still trapped inside a single cell. This is where cytokinesis comes in. It's the physical division of the cytoplasm, and there is a core mechanical difference here depending on the type of cell. In animal cells, the cell membrane is flexible. It basically just constricts, deepening inwards around the middle until it literally cleaves the cell in two, like tightening a belt. But plant cells, they have a rigid outer cell wall. They can't just pinch inwards. Instead, they have to build a whole new wall from the inside out. A cell plate forms right between the two nuclei and grows outward until the cytoplasm is totally separated. To put that into perspective, take a look at this. On the left is the plant cell, with that rigid cell plate forming dead center and pushing outward. Now contrast that with the incredibly stunning scanning electron micrograph of the animal cell on the right. You can vividly see that flexible membrane constricting and pinching deeply inward. Keeping this specific visual distinction in your head is going to be incredibly helpful for grasping these cellular mechanics for your exams. Section 4. Onion root squash experiment. Theory to practice. So, how do we actually see these cells actively dividing in a real-world lab setting? Well, we use a specific squash preparation, usually with onion root. We cut just the terminal 2 to 3 millimeters of the root because that's where the growth is most rapid. We tease the tissue apart, and then, this is the crucial step, we apply a stain called acetoursane. You absolutely need this specific stain because it actively binds to the chromosomes, which makes them pop under the microscope. Finally, we squash the tissue firmly under a cover slip to get a nice single layer of cells and apply some gentle heat to set the stain. And here are the results. This perfectly maps all that textbook theory we just learned to actual reality. In this photo micrograph of the stained onion root tip, you aren't just looking at abstract diagrams anymore. You are looking at real living cells frozen in time. You can clearly spot a cell up there in prophase with dark, thick chromosomes. There's another one in metaphase lined up perfectly down the middle, and over there, one in anaphase actively being pulled to opposite ends. It's pretty amazing to see it for real. But all of this brings us to a really crucial question. Mitosis is, at its core, a cloning process, right? It ensures every resulting cell is perfectly genetically identical to the parent cell. So if life relied solely on mitosis, we would all literally be exact replicas of each other. How then do organisms create the kind of genetic variety that's actually necessary to adapt and survive in a constantly changing environment? Which leads us to Section 5, the stages of meiosis, reduction, and variation. To answer that question about variety, we have to shift our focus to the sex organs. This is where a totally different type of division happens, one built entirely around reducing the chromosome number and generating that much-needed variation. Let's look closely at the first meiotic division. Unlike mitosis, where chromosomes act totally independently, in prophase I of meiosis, homologous chromosomes, meaning the matching chromosome you got from your dad and the one from your mom, actually physically seek each other out. They lie side by side in a process known as pairing. And it's during this intimate pairing that non-sister chromatids can physically exchange genetic segments. This is called crossing over, and it literally mixes and matches parental traits. Now let's compare the two halves of the meiosis process. The first meiotic division does something really unique. During anaphase 1, it pulls the entire homologous pairs apart. It separates the maternal and paternal chromosomes, cutting the overall chromosome number completely in half. Then the second meiotic division kicks in. This second phase functions a lot like the ordinary mitosis we just learned about. It steps in to split the remaining sister chromatids right at the centromere. And the final result of all this? 4 completely genetically unique haploid cells. Section 6. Grasshopper testis squash experiment lab check 2. Just like we observed mitosis in those plant roots earlier, we can observe meiosis by preparing a squash from the reproductive organs of a male insect. Now, this requires a slightly different approach in the lab. First, we dissect an etherized grasshopper to locate and extract the yellow testis masses from its abdomen. And here is the major difference from the plant root experiment. We must vigorously wash this animal tissue in a solvent called acetic alcohol. This is non-negotiable because it dissolves all the surrounding fatty materials that would completely ruin our view into the microscope. Once it's cleaned, we again use our trusty acetoursane to stain those chromosomes and squash the tissue flat onto the slide. If you look at this dorsal view of the grasshopper, you can visually pinpoint exactly where those yellow masses are located, right near the middle of the abdomen. The diagram really reinforces that critical step of shaking the extracted tissue in the glass vial of acetic alcohol before you even think about applying the stain. Following these exact DCN-Leia lab steps is what allows scientists and students to observe actual homologous chromosomes pairing up in real time. So, if we want to distill everything we've covered today into one single foundational concept, it's this. Mitosis is all about preservation. It duplicates the genome perfectly for growth and for repair. Meiosis, on the other hand, is about having the genome, reducing it down to haploid gametes, and totally mixing the genetic deck. That way, when fertilization finally occurs, the normal diploid number is restored in an entirely new, completely unique organism. I'm going to leave you with this final, honestly mind-boggling thought. Because those homologous chromosomes align totally randomly during meiosis, a process called independent assortment, a single human can produce over 8 million unique gametes. When you combine that with a partner and factor in the literally infinite genetic swapping from the crossing over process we discussed, the mathematical possibilities for genetic variation are easily in the trillions. Every single resulting organism is mathematically unprecedented. There is no one else exactly like you. Thank you so much for joining me for this explainer, and hey, keep questioning the microscopic world around you.